Good morning. Hello, everyone. My name is Kirsten Matthews. I'm the Baker Institute Fellow for Science and Technology Policy and Biomedical Policy. Welcome to Rice University. Welcome to the Baker Institute for Public Policy. It's a pleasure to have you here today for our event, The Road to the Nobel Prize, with our guest speaker, Dr. Allison from MD Anderson. This morning's discussion is sponsored by the Baker Institute Center for Health and Biosciences. This center is actually run by Dr. Vivian Ho, and we use evidence-based research to promote policies that impact health and well-being of our population in Texas, the United States, and globally. We have 4 major research themes right now that include health economics, which Dr. Ho runs, biomedical research, the program I work with, maternal and child health with Dr. Kianta Moore, and control of neglected infectious disease with Dr. Peter Hotez. Today's program is actually a collaboration with the Swedish American Chamber of Commerce, who are here today, thank you, as well as the British Consulate, with our Consulate General who's here with us today, Ms. Astrid Markland as well, and with the company Miraculum, with Dr. Anne Christine, who's here with us. In addition, we would like to actually thank our Health and Policy Forum donors. If you're interested in participating in this program, there's information on the back of the program of who those are. So I'm not going to spend too much time here because I think everyone is more interested in hearing Dr. Allison talk. Dr. Allison, as we know, is part of UT, University of Texas, MD Anderson Cancer Center. He's the chair of the Department of Immunology and the Vivian L. Smith Distinguished Chair in Immunology, as much of several other different titles he has. He as well is the 2018 Nobel laureate, or one of the Nobel laureates for medicine. So with that, I'll have Dr. Allison go.
Okay, thank you so much for that kind introduction. I'm really honored to be here at the Baker Institute and at the Swedish American Chamber of Commerce. The title, I don't know, I wouldn't have used that one, but it sounded good. It was the one that was on the program, so I decided to use that. And what I would like to do is really talk to you about how we went from basic science to a new way of treating cancer. Really, I'm going to take you through the history of it a little bit and then try to show you some current work that we're doing, that we're in the middle of, trying to extend the efficacy of this new approach of mobilizing the immune system to attack cancer, to subcancers that aren't currently responding very well. And then talk a little bit about the future. Then I'll be glad at the end to talk as long as you want to, answer any questions or discuss any topics you'd like. So I'd like to start with this. This is Sharon Belvin, the first patient I met. She was diagnosed in 2004 with stage 4 melanoma. She was 22 years old, had just finished college and just gotten engaged to be married. And she had, very, very little on the outside of her body, but she had brain mets, she had liver mets, she had all of the inside was just riddled with metastases, and she failed every therapy they gave her. And so it's sort of a Hail Mary throw. They put her onto a phase 2 trial. It was relatively new at the time, and within a few months her tumors were completely gone. And this photo was taken when she came back, and her doctor said that Jed Walchuk said, she was completely tumor-free. And Jed's stories were pretty amazing. He said he got the CAT scans back, and the people that looked at it, the pathologist said, these samples must be mixed up because there's no tumor in any of these, And so that was, anyway, this was really early in the development days. And so anyway, she went on to have a complete response, and this is 10 years later with her 2 children that she's had now, and she's still doing fine. I've gotten to be pretty good friends with her and followed her with all her experiences since that. She actually went to Stockholm with us in December to share in the glory there. Anyway, but it just shows you, what this can do. She was actually told by our doctors, though, after the therapy that she shouldn't have any children, because they were worried that there's some immunosuppression that's accompanied pregnancy, and they said, your tumor may come back. And after a while, she just said, to hell with melanoma. I'm going to live my life, and so that's what she did. She just started, lost a lot of weight, became a triathlon athlete, of course, had the 2 children, and now is living a remarkably vibrant life. Anyway, sort of thing that really touches my heart anyway. So, get back to the story then. Why immunotherapy? Well, cancer is a genetic disease, obviously. There's a lot of gene mutations, genomic instability. There's really many different kinds of diseases. It started to be classified not so much by the tissue it comes from, but by the mutations that cause it, KRAS mutations, for example, or other drivers. And this also opened the so-called personalized medicine, where people use genomic sequencing and then identified specific mutations that seem to be causally associated with the cancer, and then would try to, most of these end up being enzymes, tyrosine kinases involved in signaling pathways that tell the cell when to divide, when not to divide, when to die, when not to die, those sorts of things. So people began to then make inhibitors of those and try to treat cancer with it. And you probably heard about a lot of these. They can be pretty miraculous. The patient that has the right mutation, about 90% of the time, the tumors will basically disappear within a few weeks after treatment. The problem is, because of the genetic and genomic instability, by the time you treat the cancer, there are already enough mutations around where there's tumors with other driver mutations, and they can come back, and then you have to do it again and again. In melanoma, for example, the favorite target of personalized medicines is a mutated protein called BRAF. Doesn't matter what it is, but at last count, there were 13 different ways a tumor could escape a drug that would completely inhibit BRAF. And so it's hard to think with that situation how this approach to treating cancer, would ever really be curative. I'm not trying to say it's not valuable, because it can buy months and months to patients, but it's just not likely to ever be really curative. Immunotherapy, on the other hand, is very different. First of all, the immune system loves mutations, and it doesn't matter whether they're the driver mutations or the passenger mutations. They give rise to things that are in the cancer cell that aren't in the normal cell, and that's what the immune system looks for. It's things that shouldn't be there, be it a virus-infected cell or a cell that's got mutations. And again, it doesn't matter whether the mutation's in the BRAF molecule or in some irrelevant molecule. In melanoma, the immune system's going to treat it the same. It knows it's something that ought not to be there, and the cell that has it ought to be eliminated. And the second thing about the immune system that makes it really special is memory. Once you've got T cells against a certain antigen, a lot of them become stem-like cells that renew for the rest of your life. So the drug doesn't go away, and in fact, it's there. You've got it forever. If the tumor comes back, it can be reawakened and go after the cancer again. And finally, there's adaptability. You've probably got somewhere, nobody really knows the top number, but somewhere between 70 and 100 million different T cells that can recognize that many different things, and it's incredibly adaptable. If the tumor changes, the immune system has been shown to change with it. And so if you can just mobilize it to make it, more effective, then it's equally a match for this genomic instability that occurs in cancer. And so immune checkpoint blockade, which is what we developed, was really a paradigm shift in cancer therapy, in cancer therapy and immunotherapy, really, because it doesn't target the tumor cells, as I will show you. It doesn't involve vaccinating or with anything or doing anything to turn on an immune response. It works by blocking inhibitory pathways that are keeping T cells from doing something they would do anyway, and unleashes anti-tumor responses. And just to kind of illustrate this, this is the whole basis of this way of treating cancer, and it came from, not from trying to cure cancer. It had nothing to do with cancer. It came from basic science, from fundamental studies of fundamental mechanisms involved in T-cell biology, basically. And it just turned out that some of the lessons were useful for developing new strategies for cancer therapy. And so the story sort of starts here. When I was a graduate student, a professor I had for an immunology course said, he was an antibody guy. You know, those are made by B cells. I think everybody knows that. But there were these T cells, these new cells that had just been discovered. And, you know, he talked about them and said, they go all over your body and look for stuff that oughtn't to be there and then deal with it. And I said, you know, gee, how does that work, you know? And went to talk to him. He said, well, I don't know. I don't think anybody knows. I said, I don't even think there's such a thing as T cells. I think some other cell just picked up an antibody molecule and hijacked it and go and use this working anyway. I thought, well, I don't know. I think I'm going to work on this. And so that was in 1968, I think, something 7 or 8. And so I'm still working on it and haven't quite gotten, still a lot to learn, and we're still finding out anyway. So anyway, one of the first things I did in 1982 out at Smithville, at the MD Anderson lab at Smithville Science Park, worked out the structure of the T-cell antigen receptor, which had not been known until that time. And that's the structure, it's like the ignition switch in a car. That's the thing that the T-cell uses to know that something's foreign. You know, that's the sensing molecule that tells you something ought not to be there. But by the late 80s, we knew that that wasn't enough. There was another signal. You know, it's not just flipping a light switch. There's another signal that people called a co-stimulatory signal. Biologically could be shown had to be delivered at the same time as the antigen receptor signal in order to really turn a T cell on. And so in 1988, we showed that that signal was provided by a molecule called CD28 that binds to specialized molecules that I'll talk about more in a little bit that are found on professional antigen-presenting cells, a very discrete subpopulation of hematopoietic cells, of blood cells. Anyway, that interaction, the contemporaneous engagement of CD28 and the antigen receptor by their ligands, looks like the ignition switch and then the gas pedal, and you turn the car on. It starts making a cytokine called IL-2 that allows T cells to divide. And so that was the story for a while. But when we cloned the gene for CD28, there was another gene very similar to it, a molecule called CTLA4, that had already been cloned, but nobody knew what it did except that it was in activated cells, not in resting cells. So it didn't appear until after the T cell got the T cell receptor signal and the CD28 signal. Anyway, there's a lot of controversy about it. A lot of people said it was another CD28, another gas pedal. But we actually showed in 1994 that it was actually an inhibitory molecule that turns T cells off. And this is of critical importance in several ways. I mean, one of them is the first thing that happens when T cells get activated, you've got 100 billion, which means you've only got a few of any given specificity. They're clones. And so you turn the clone that sees the thing that, you know, that it's going after, and you've got to get a lot of them really fast to get, you know, the troops in the field and go out and kill the virus-infected cells or the cancer or whatever, and you need hundreds of thousands. And so when the T cell gets the T cell receptor signal CD28, they start dividing. They start dividing every 6 hours or so, as fast as bacteria, sometimes to generate, you know, the protective army that's needed. And then they differentiate it, they start, you know, dealing with the problem. But you have to stop that, because if you don't stop it, it'll kill you. And that's what CTLA4 does, and we showed that in several other labs by inactivating the CTLA4 gene. And when you do that, mice fill up with T cells. Their immune response can't stop, and so it's lethal. And so that was, you know, that was a clue that it limits responses. But actually, we started our tumor work even before we absolutely knew that. But that's the biology. It's involved. It's hardwired in every immune response, because you've got to stop that proliferative phase and let the T cells move into a more quiet cell phase where they're just going around and killing stuff or making whatever they need to make. So again, it's complicated. But one of the things we figured out after a while was that T cells can recognize antigens on tumor cells through the TCR, but they don't get activated because tumor cells do not have the second signal that binds to CD28. And that's thought to be a way of maintaining, avoiding autoimmunity. So because of that, the tumor cell is invisible to the immune system. It may have antigens, and we showed this experimentally by taking tumors which would grow and kill a mouse, and we just put the gene for these B7 molecules, which is the thing that binds CD28, and the tumors immediately reject it. So it's not that they don't have antigens, they just can't fully activate a system. So what happens, the only way you get an immune response started is for some tumor cells to die, either because they get too big and they can't get enough sugar or oxygen or whatever, or radiation or something anyway. But for tumor cell death to occur, it causes inflammation then, and that causes these antigen-presenting cells to come around. They pick up the dying tumor cell bits, redisplay it on their surface, and they've got these B7 molecules, and so they can give both signals. And then that tells the T cell that there is a tumor and you've got to do something about it. And then that, again, activates so they make IL-2, and that tells them to start dividing. And so, there you go. But as with anything, that also turns on CTLA-4 after a time, and then it can just stop that process. And so it's kind of like a clock, and so we think it's just a race. The tumor's invisible for a while, and then the T cells get turned on, we get tumor cell death, and then it's a race. If you get enough T cells before CTLA-4 turns the process off, you kill the tumor. But if you don't, then the tumor wins. And so I had this very simple idea: just turn off the brakes for a while, just block CTLA-4. And so it's a cool idea for a number of reasons. One, again, you're not treating the cancer, you're treating the immune system. So the kind of cancer is irrelevant. It's got the possibility of being a drug that could treat all cancers. And the second thing is the whole process starts by tumor cell death. And so you could combine this with radiation or with chemotherapy or with the small molecules that I was talking about earlier that kill tumor cells, cause inflammation, and start an immune response. But you should give all those things the properties of the immune system, memory, vast access to many other targets that, by just doing combinations. Anyway, does it work? Well, this is what our early experiments, this is a tumor growing on the backs of mice. We have a number of, genetically identical mice and a number of different tumor lines that we work with. And so this just shows in the center here, the tumor, if you don't treat it, you inoculate the tumor and the tumor grows for a while, then you have to euthanize the mice. If you block CD28, the tumor actually grows faster, showing that there is a nascent immune response, but it's not affected. But if we just injected CTLA-4 antibody, the tumor grows for a little while, then gets rejected. And the mice are permanently immune. You can challenge that mouse for the rest of his life with tumor, and they'll just shrug it off. They will not grow. And so we did this with many different kinds of cancer and many different kinds of mice. And it didn't always work. It worked. This tumor was a chemically induced tumor. It was very immunogenic, call it. So very immunogenic tumors, CTLA-4 blockade would work by itself. But we had some spontaneous tumors that were very poorly immunogenic, and everybody thought that they were, up until the time we did this work, thought that you couldn't treat them. But we found if you add radiation or you add a vaccine, we could deal with those too. So we really never found a mouse tumor that we couldn't treat successfully with either CTLA-4 or some combination. And so we worked for about 3 years. I was talking about this someone earlier. For about 3 years, we tried to get somebody to help us take this to the clinic, and we ran into a lot of problems. The people have been thinking about immunotherapy for decades, and it's never worked, so it won't work. Or anybody can cure tumors in mice, but it's not going to work in humans. Or blah blah. How can you treat cancer by ignoring the cancer? Well, I don't know. I'm Buddhist, I guess. I think it's Zen. You ignore the cancer and treat the immune system. Anyway, so finally we found this company named Metarex that had this cool technology where they had mice that had their antibody genes removed and replaced with human. So we could make in those mice a human antibody to CTLA-4, which is important because if you make a mouse antibody, you inject it into humans, it'll be recognized by the patient's immune system and reject the drug. And so this is a way of getting something ready to go. And so so this is a fully human antibody, and ended up having a lot of effects I'll talk in more detail about. But I'd like to just tell one story. When we started on this, we talked about the MDX project, James Bond, or this was the antibody 10, so it was MDX-010 were the ones we talked about, that 010 project. And then the FDA named it ipilimumab, which was really depressing and disappointing to everybody, it's such a squeegee-like name. And so anyway, I tried to convince the head of Medirex to go to the FDA and tell them to put an H in front of it at least, since it came out of Berkeley, but they decided that ipilimumab would not be enough gravitas for a cancer drug. But anyway, the rest of the newer ones have names that are equally as bad. But anyway, IPI, as it's called now, in early trials had objective responses, just as we would have predicted, to many different kinds of cancer, including melanoma, prostate, kidney, bladder, ovarian, lung, etc. We thought it would be completely free of side effects. We had never seen any in the mice or in monkeys, where we did a lot of safety testing. But it turned out in people there are adverse events which can be fatal if they're not dealt with, including colitis, hepatitis, apophysitis, pneumonitis, a lot of itises. So these are inflammatory reactions that are sort of immune responses run wild. But they're really not autoimmunity, and they can be dealt with most of the time, but with systemic steroids and whatever, and you can wean the patient off the drugs and they go away, don't come back. So it's manageable, by a physician who pays proper attention. I should add, though, that recently it's been found now that hundreds of thousands of people have been treated, that there are people who develop type 1 diabetes, although they may have had subclinical disease first. And there's a very rare incident of a T-cell mediated myocarditis, a heart problem, that are legitimate autoimmunities, but they're quite rare, actually. Most of it are just these temporary misbalances. So this is my next favorite slide. This is a woman also named Sharon, actually, but she was in LA, and she was diagnosed, again, stage 4 melanoma, and had failed everything, including 2 immunotherapies, did antigen pulse dendritic cells and high-dose interleukin-2, and progressed right through them. And so Tony Rebus, her doctor, said, there's this new drug, it's never been in people before, and I've got a trial of it, phase 1 trial, and if you'd like to try it, we could try it. She said, I'll do anything. I need about 3 months to see my son graduate from high school. And so her tumors completely went away. I visited Tony in 2011, and she had just had, showed on the right, that is her CAT scan 10 years after that. So, and she's still doing fine. So she's 18 years out now with only 1 injection of antibody, with no further treatment at all. And so, these 2 ratios show the durability of these kinds of responses. In any event, so this is a phase 3 trial, registration trial. This is a placebo control, which we don't do anymore. It was done then because there was no drug that had ever, in any clinical trial, extended life with melanoma. So there's no phase 3s comparing the drug to the standard of care. There was no standard of care for melanoma because there were no treatments. There were treatments, but they'd never been shown to be effective. The median survival after diagnosis was between 8 and 11 months. So it was essentially a death sentence for about 98% of patients. A couple of percent would make it, but you can see this is the placebo line. This is the ipilimumab curve. So you can see it moved the median survival over about 4 months, and that would have been enough by FDA standards for approval. But the really cool thing is that if you follow the curve down, in about 3 years, it flattens out. A little over 20%, it stays there for the almost 5 years that it took to do this trial. And so it was approved by the FDA in 2011, the first drug in this class. And so a couple of years ago, there'd been enough patients, about 5,000 patients in this study, retrospective study, where there was 10 years follow-up. And you can see here that, again, you see this inflection point at about 3 years, and it flattens out. And what that means is essentially nobody dies of melanoma after about 3 years. And so if you make it 3 years, you're good to go. I mean, 10 years was just a handy, endpoint because they had the data. So, it confirmed that tail on the survival curve that was observed in the phase 3. So anyway, that's it. So for IPI now, it's out there. It'll come back to where it's approved in a little while. But anyway, so the question now, of course, is, well, what about the other 80%? Why didn't it work in everybody? And there's some mechanistic reasons for that, perhaps. If there's not this dying and priming, dying of tumor cells and priming of T cells, if that's not happening when the drug's on board, maybe it doesn't work. But of course, a more interesting possibility is that maybe there were other checkpoints, because our work showing CTLA-4 was a cell-intrinsic inhibitory mechanism. That was the very first one ever found in the immune system. But it turns out there's another one called PD-1 that had been known. The gene had been cloned many years before, but nobody knew what it did. But after our clinical work came out in 2001, Tasuku Honjo, who had cloned the gene, who shared the Nobel Prize with me, and a group at Dana-Farber Cancer Center, led by Gordon Freeman. Gordon's lab showed that there actually, PD-1 has two ligands, also PD-L1 and PD-L2, and it's a little bit more complicated system, but what they showed was that it's a little bit different than CTLA-4 because one of the ligands is actually expressed on tumor cells. And the model is that the T cell gets activated as an activation marker, puts up PD-1. T cell makes the cytokine called gamma interferon, which can kill tumor cells, but the tumor cell can respond to that by upregulating PD-L1, the ligand for PD-1, and when that interacts with PD-1 on the T cell, it turns the T cell off. And so this is a tumor acquired resistance mechanism, and it's been shown that this same pathway plays a role in protecting a developing embryo from attack by the mother's immune system, seeing paternally encoded molecules. So this is a normal pathway that tumors just seem to have picked up to protect themselves. In any event, PD-1 quickly went into clinical development, and this was a phase 1 trial. It was reported in 2012. Good response rates in melanoma, non-small cell lung cancer, renal cell, kidney cancer. There were no responses in colorectal carcinoma, although it's been shown since then that there's a subset of colorectal cancer that has defects in DNA damage repair, so it has a lot of antigens, and so that does respond. And there are also no responses in castrate-resistant prostate cancer, which is a surprise because, as I told you, prostate cancer did respond to ipilimumab, and so we've figured out what that is, and I'll show you why that is in a few minutes. Anyway, where do we go from here? Well, obviously we've got two of these checkpoints to start with different mechanisms, so maybe you can put them together. And so this shows a trial that was originally carried out by Steve Hody and Jed Walchuk. This again is IPI by itself, but if you add PD-1 antibody nivolumab to it, the response rate goes up to over 50% in this short-term trial. And there was recently a 4.5-year follow-up published, and it's still about 55% or so. So that's pretty amazing because it's made it past, this is short, but like I said, the 4.5-year data shows it's still up there. And so if that's correct, and if it lasts for 10 years, as IPI did by itself, then maybe that's going to be the new benchmark for our ability to treat late-stage melanoma. And so, again, we started this work, there was no therapy, and the median survival after diagnosis was, as I said, just several months, less than a year. And so now it looks like maybe 55% or so of patients are going to, be good to go. Maybe considered cured. We'll see. It's not out there yet. So where are we? Well, this is a partial list of what's going on. First of all, there are a whole lot of different antibodies to PD-1 out there. They all seem to be about the same. There's also antibodies to PD-L1 in trial. But as I mentioned, ipilimumab was approved for melanoma, and then PD-1 antibodies, a couple were approved in 2014. And since then, there's just been a deluge of approvals by the FDA. It may be obvious to all of you, but that's an important milestone, FDA approval, because it means any doctor can prescribe the drug. It's no longer experimental. And more importantly, the insurance companies have to pay for it. So it's quite expensive, more expensive than it should be. Anyways, with all these things, we start off with late-stage patients that don't really have any other choice. You develop a drug, and then it moves earlier and earlier. So now these are approved also for adjuvant melanoma. So in other words, for people who had localized disease, had surgery to remove it, and then they get, so they got minimal disease, and they, were treated very early, and so now you can treat them, and it's effective there too. So it's not late stage anymore, and it's the rest of these are gonna start moving earlier and earlier too. But anyway, also approved for pediatric melanoma. Anyway, non-small cell lung cancer, also small cell lung cancer recently, renal cell cancer, Hodgkin's, bladder cancer, head and neck, Merkel cell, which is a skin cancer. It's even more lethal than metastatic melanoma. Stomach cancer, liver cancer. There's more, coming all the time. As I said, I don't know, I probably missed 10 or 15 approvals in individual states, but it's really expanding. Again, all of this since 2014. 2011 actually was CTLA-4. And one thing that's really cool is this is the first approval for a cancer drug based on a genomic defect rather than the tissue. So this is for patients with defects in DNA damage repair, microsatellite instability. Anyway, this just means that they're going to have a lot of antigens for the immune system to attack. So it doesn't matter what kind of cancer you have. If it has defects in DNA damage repair, it's a candidate for being treated with these PD-1 antibodies. So anyway, it's really fascinating what's going on. But we really need to understand mechanisms, because there's so many combinations now. Just among these drugs, there's a lot of combinations, but then you can combine them with radiation. As I said, you can combine them with certain chemotherapies. You can combine them with small molecules. There's more checkpoints now, and I'm not going to get into specifically until right at the end. But there's so many now, so many combinations that there's something like 3,000 trials of PD-1 antibodies against plus PD-1 plus something, and that's exciting. But it's really wasteful the way it's being done, because people just set up a small clinical trial, look for a clinical signal. If they don't get one, they move on. And I think that's really criminal, because we know now from really studying tissue samples for patients before treatment and then on treatment, and then people that respond or don't, we've got a pretty good idea of what the successful response looks like. And to have a failed trial, a trial that missed its clinical endpoint, and not learn something about what happened, I think it's almost criminal, and should not be allowed, because it wastes patients, wastes drugs, and wastes time. Very important. So anyway, along those lines, we started at MD Anderson what we call the immunotherapy platform. Pam Sharma, who's a genitourinary oncologist, and I started this with strong support from the Anderson administration. And the whole goal of the immunotherapy platform is to carry out trials, but in a way that we get blood and we get tumor cell samples, we can study them, take them to the laboratory, see what's going on, and very quickly then go back to the clinical trial. So very rapid iterative interactions from clinic to lab and back and forth. And it's been pretty successful so far. We're participating in a little over 100 trials now with 18 MD Anderson departments, about 3,000 patients enrolled, and many samples studied. So there's a lot of stories I could tell you, but I'm going to focus on prostate cancer today. So as I told you, there were no responders in the phase 1 trial of PD-1, and there had been with anti-CTLA-4, but when BMS, Bristol Myers Squibb, went to a phase 3 trial, this is the results from that trial, the CTLA-4 ipilimumab failed, to show a real significant difference from the standard of care. And so it works in some patients, but the trial failed. So they shut their whole program down and said, it's not an immunogenic tumor, it's not going to work. We have a pretty good relationship with them, though, and if we go to them with an idea, they've been pretty nice about giving us free antibody to do small trials with. So I'll show you in a minute, but something just to put out there. With the advent of genomic sequencing, it's become clear that cancer is a genetic disease, but different cancers have different mutational burdens. Up here you can see melanoma, melanoma is caused by UV radiation, mutations caused by UV radiation from sunburns, and then lung cancer, these two here, of course, caused by mutagens in tobacco, and so also to some extent stomach cancer, throat cancer, bladder cancer, and colorectal cancer, all caused, at least in part, by mutagens in tobacco. So those all have really high mutational loads that respond very well. So there have been approvals to all of those types of cancer because they respond well, which is really kind of ironic because the high mutational load is what made them resistant to chemotherapy and made them not very good targets for the personalized things because they were, but again, that makes them sitting ducks for the immune system because the immune system loves mutations. You come down here, and there's sort of a fall off, and kidney cancer is at a level that it really shouldn't respond because it has so few mutations, but actually it does respond quite well. But you come down here and there's pancreatic cancer, and we haven't gotten very far with pancreatic cancer. We're trying. Glioblastoma's down here too, but prostate cancer is pretty low. And as I said, it might not even be expected, except that there were responses to CTLA-4. So, in addition to the existence of antigens, there's another issue, and that's the state of the immunogenicity in the tumor. Many tumors like melanoma are really filled with T cells, macrophages, other elements of the immune system. So all you have to do is take the checkpoint off of those guys and you can get a response. But other tumors may not have any infiltrates in them. And so if checkpoint blockade's going to work, you've got to get T cells into those tumors. And so we then wanted to take a look at prostate cancer, and this was a trial that Dr. Sharma carried out, where patients that were going to surgery, had localized disease, were going to surgery anyway to have they were going to be cured by prostatectomy most likely. But she set up a system where she gave them a couple of doses of anti-CTLA-4 before they went to surgery. And so we got the entire organ to study. That's enough tissue to do all the sorts of assays that we wanted to do. And so this is what we found. I hope this is visible. As you can see here, we've stained, and the positive stain is a brown color. But you can see before treatment with anti-CTLA-4, these tumors are very cold. No CD4 T cells, no CD8s, no regulatory T cells. It's a general immune marker. Some other molecules. This one's on CD8 T cells that kill cells. These are all macrophages. So nothing really going on before. But after treatment, you can see robust infiltration with all those cells bearing all those markers. So then it looks just like melanoma. Before it looks like pancreatic, and after a couple of doses of anti-CTLA-4, it looks like melanoma. So anti-CTLA-4 can drive T cells in, and PD-1 doesn't. I can just tell you that right now. I don't have time to talk about it, but these are really very different drugs, CTLA-4 and PD-1. But when we looked at the genes that were expressed, well, we had infiltrates now, But these are all negative checkpoints. You know, you get PD-1, PD-L1, another molecule called VISTA, another one called LAG3 actually. So we've gotten T cells in, but it's also induced all these compensatory things. It's made all these checkpoints come in. So we think what's going on is, you know, before CTLA-4 there was nothing in there. You give CTLA-4, the T cells are there, but they've got all these inhibitory pathways. Here you can see PD-L1 in the immune cells in the tumor, and this other molecule called VISTA that's very recently discovered that we've been working on for a while. And this just shows you the PD-L1, it's on the tumor cells, it's on a subset of macrophages, actually it's on the T cells themselves. So this is a very, very suppressive environment. So anyway, we went to Bristol Myers Squibb and said, look, what's the solution? This is obvious. You give CTLA-4 to get the T cells in, you're going to get these negative pathways, and then you give PD-1 to block those pathways. And let's see what happens. And so Dr. Shorman designed this trial, and this is in castrate-resistant. This is late-stage prostate. And the patients go in and they get 4 doses of the combination, then go to maintenance with just anti-PD-1. And this is one of the patients here. Several of them responded. You can see this guy's PSA was going up, went up to over 400. After the first dose, it went down to 300. After the second dose, it went to undetectable and stayed there. All this guy's metastases went away. I actually ran into his doctor yesterday at Anderson, Chris Logothetis, and this guy was going to the Bell ceremony. You know, he's finished his therapy, and he's completely tumor-free now for 2 years after this. So this has led then to another trial, a multi-institutional registration trial that should start any day now. We're hoping that this will, one of the limitations of this trial was the dose of IPI was a little high, and so there was a lot of toxicity. It's based on melanoma dosing, and prostate cancer patients are older and sicker typically, and they can't take the harsh side effects that a melanoma patient can. So we've got to dial that down a bit, and so that's happening in the new trial. So we hope that this is going to prove successful there, make prostate accessible. And then this is just something that I want to show that we're still working on. So I'll talk to you about neoantigens. Well, prostate cancer has a very low mutational burden, as I told you. So one of the questions was, are there neoantigens in prostate cancer? So what we did was take patients, we resected their tumors, we did DNA and RNA sequencing to determine what the mutations were, and then did appropriate assays, as I'll show you, to see if any of these actually provoked immune responses. Also looked for immune responses to shared antigens, prostate-specific antigen, prostate-specific membrane antigen, and prostatic acid phosphatase, all 3 of which are pretty specific for prostate cancer. Anyway, the patients then went to anti-CTLA-4, and then just before each dose, we took blood to look for assays. Anyway, so there were responders in this. You can see there's responders here, non-responders here. The blue lines are the responders. You can see several of these are ongoing. So again, in some patients it was effective. And I'm going to talk about the greens are ongoing. So I'm going to show you patients 5 and 7, which had very interesting responses. So these are the number of mutations in all the patients. So patients 5 and 7 had well, the only ones that matter are the ones that are actually expressed as RNA and proteins significantly. And so this was 12 for 5 and 10 for patient 7. So very small number. And I'm mentioning these numbers because in melanoma, it can have 3,000 mutations, and maybe 3 of those will end up producing an immune response. So, you know, a lot of people said, well, if you have a low mutational burden, there's no sense in counting on those. So anyway, we then looked, and this patient 7, for example, this just shows T cell assays. The dark spot is the way is indicative of a T cell that's seeing antigen. So this is the wild type. This is the mutant that we identified down here for these 2 proteins. It doesn't really matter what they are, but this one has a mutation here at this position, and this protein has a mutation here where's the M at this position? In any event, so what we did was compare the response of the T cells of these patients with the wild type versus the mutant, and what you can see is the wild type, there's no response. The mutation, there is a response to these two proteins. So there are these two bona fide neoantigens out of the only 12 mutations that this patient had. These are controls. These are responses against viruses that everybody's been exposed to, and there's a negative control, HIV peptides. But anyway, that's against the mutation and the wild type. So both of those, there weren't any responses till we gave anti-CTLA-4, but there were afterwards. So might these have been responsible? These were responders. Might these have been responsible for this response? Well, we can't tell really, because many of the patients, also the dark things here, had responses to these shared antigens, PSA, PSMA, and PAAP. PSMA probably isn't telling you anything worth knowing, because you get T cells against that in patients who don't respond as well as patients who respond. But these two, PSMA and PAP, we only saw responses to those in patients that had clinical responses. So, we can't say that the mutated antigens were the targets, but anyway, it gives hope for the fact that they could be, and anyway, but the shared antigens are there for the immune system to see as well. So one of the things this means is it would be possible, in the case of prostate cancer, maybe to make a vaccine consisting of those peptides that the T cells are seeing, and just boost up the response by immunizing them with a combination of those peptides, in addition to giving anti-CTLA-4. So I think just going to sort of start winding it up. So where are we? Well, now we're at the point of blocking multiple checkpoints, negative and positive. Didn't have time to tell you, but we found another molecule called ICOS that's expressed, that's induced on T cells in patients treated with ipilimumab. It's a positive activator of T cells and makes them give a stronger response. And so we've made formed a company, have to have give a disclosure, and we're doing trials now to see if that might be an effective target. So in the future, there's going to be, as I mentioned earlier, a combination with conventional therapies that are appropriate, because they think there's already a lot of data. There's not too much activity here, but a lot of data in mice that we generated and others, that if you kill a bunch of tumor cells, you can make the responses better by adding immunotherapy. And also radiation, so both chemo and radiation. And one of the nice things about it is that, particularly in radiation oncology, the practitioners are now realizing that they can change the way of what they've been doing. Because with chemo and radiation, one of the problems is the side effects, because they're trying to kill every last tumor cell with the radiation and with the chemo, so they give them dose and dose and dose and really high dose, why people get sick, and it doesn't usually work anyway. But here they don't need to think about killing every last tumor cell. They can just kill enough to get the immune systems notice, and then follow it with immune therapy and perhaps do it. And one group at Anderson, and also another group at Cornell Weill in New York City, have a very good success in melanoma, lung, and breast cancer with that. Also targeted therapies, there's beginning to be some work done with the small molecules I was talking about. And there is some work now, it's early stages, to do the sort of thing that I was telling you about, identify the neoantigens, the peptides that you could then vaccinate a patient with, and enhance responses to the patient's own antigens that are relevant. And this hairball here is not meant to make any specific point, except that there are a lot of other molecules that that can be brought to bear on cancer, because the immune system's got a lot of different activities, and you've got to regulate that to make sure it doesn't get out of hand. And I doubt if any of these newly discovered ones are going to by themselves be knockouts. I think with the experience that we have and everybody else, I think where we're moving towards is the combination of anti-CTLA-4 and anti-PD-1 is going to be the core immunotherapy in various ratios, with additional ones depending on the cancer. VISTA, for example, I showed you was in prostate cancer. You don't see VISTA ever in melanoma, so you wouldn't want to use that. But if you added it to the PD-1 and CTLA-4, that might make, assuming that combination works in prostate cancer, that triplet may make it work, even better. And so, it just takes science. It takes hard work to really, look at the tissues, find out what's there, and, do rational combinations that'll bring things together that'll be additive, not do anything or even cancel each other out. So we need to improve the way that we're doing this stuff in the field. Anyway, I always like to close by saying, this is what we've been used to from the era of chemotherapy for the last 30 years or so is, do 1,000 patients and look at the median survival. And if you get a few month increase in that, call it a success, which is nothing wrong with that, certainly. I mean, it's getting any survival benefit's really good. But we know from ipilimumab that we can do that and get a 20% or so cure rate, it looks like. And so in the future, what we need to be doing is moving that up, not worrying about moving this over so much, but moving that tail up with the proper combinations and doing it as many kinds of cancer as we can. And it's still a daunting challenge, but the good news is we know the basic rules now, and we know the sort of things to look at with a little planning and a lot of very expensive instruments and people working on this. I think that we've got a chance of success, at least in some cancers. Like I said, it's disappointing in pancreatic and glioblastoma, for example, but we're beginning to see some chinks by studying the tumor, and the individual properties that they have that make them resistant, and we're beginning to know what those are and can begin to attack them in a more rational basis. So with that, I'll stop and take questions if anybody's got any energy. So thank you for your question.
So I'm Christine Langselius. Thank you for a very good presentation. How do you determine what is just enough of the cells that needs to be killed in order to right now where you reach the 22% level?
Yeah, you mean with radiation or something? It's by experimentation. What Sylvie Fournier, for example, at Cornell Weill is doing is, first of all, trying to work it out so that she's not killing immune cells. Because what happens is, she realized this some time ago, you give a beam of radiation, you kill the tumor cells, that causes inflammation. The immune cells rush in, those antigen-presenting cells especially, and start priming an immune response. Then you give another dose of radiation, you kill them. And so first of all, you got to give the doses fractionated away to avoid that destruction, and then you just do it by looking and seeing what works experimentally. But she's found it's really a small fraction of the total cells. I can't tell you what that is, but she knows the dose, beginning to know the dose and how to fractionate it.
Very good, thank you.
It is just super interesting. Thank you very much. I was a little despondent to hear that you haven't found anything for glioblastoma yet.
Yeah.
I thought Jimmy Carter got immunotherapy for glioblastoma. Is that not correct? I know that's a fast-growing cancer.
What did you say?
The President Carter, didn't he get some immunotherapy?
He had a brain cancer, but he had melanoma.
Oh, okay, it was melanoma. I thought it was a glioblastoma.
So melanoma in the brain responds just as well as melanoma.
So that explains it. I thought it was a glioblastoma. Okay.
No, glioblastoma I have trouble with. But it's not. Anyway, the point from that is that it's not just that it's in the brain, because melanoma works fine, but there's something about glioblastoma and one thing we know is it has a lot of macrophages in it that it can be suppressed. They can suppress T cells, and maybe we can do something about them.
Thank you, Dr. Allison. My question is, what kind of a success rate have you had with immunotherapy with hematological cancers?
Yeah, that's a good question. It's just beginning, really. In Hodgkin's lymphoma, the response rate's about 90% with PD-1, and that's because in Hodgkin's there's a translocation, an amplification of a gene segment that has the PD-1 and the PD-L1 molecules on it, and so they express high levels. And they must have an antigen coming from somewhere. Nobody's identified it yet because that in and of itself wouldn't work. So there, and AML is beginning to show some responses. Others are just coming along. It's not been as successful as with solid tumors, but it's early days yet.
Thank you. So today we see amazing effects with monoclonal antibodies to the regulatory proteins. What do you think the prospects are of replacing these costly treatments with small molecules?
I think that people have tried that a lot. We tried working with a company to make aptamers, these RNA molecules that are relatively small, and just couldn't make one that would block. I think that a lot of protein chemists that I've talked to say the surface area that's involved, and these contacts are just too large to be, blocked by a really small molecule. I don't know if that's true or not. But on the flip side of that, these things are all really expensive, but it's not because the antibodies are expensive to make. Because right now, these that are well along in clinical development are usually made in yeast. So it's like a byproduct of beer, almost. You can pull the protein out of the culture. So it's not hugely expensive to make. It's just, the other things associated with the pharma industry and charging. And I don't want to get involved in the middle of this argument, but, I saw what Bristol Myers Squibb went through with anti-CTLA-4, because it was the first in its class. They had to change the endpoints. They had to change the way the trials were done. It was all new, and it was their trial. Most trials take 18 months. Their trials took 5 years. So it was hellish expensive for them. But, okay, that was almost 10 years ago now. So at some point, they've got to have been paid back. But anyway, like I say, that's not my job description to argue about that one. But it's not the cost of the drug.
My name is Salem. We're across the street from you, Bertner Avenue. And I'm a medical oncologist. I've been 51 years in cancer medicine. And I want to thank you for what you have done to cancer medicine. I think that's the most major leap in cancer medicine over the last 30 to 40 years. And I'm probably one of the beneficiaries of your work, as we have recently tried to combine, as you had recommended, immunotherapy plus chemo, standard, plus targeted treatment. We have treated a total of 19 patients. We submitted an abstract to ASCO, which has been approved for publication. And we were very impressed by the response of the 19 patients of very advanced refractory cancer, different types. We had 11 complete remissions and 7 partial remissions. And the 7 partial remissions, 5 of them had more than 80% reduction in volume. There is something mysterious about combining immunotherapy, to me, combining immunotherapy with chemo and targeted treatment. My question to you is, do you have an advice as how we should combine these treatments?
Yes, I think that there are, like I said, umpteen possibilities of how to do that, but I think it's important to first know the impact of the chemotherapy or whatever you're using on the immune system itself, because if you're going to be shutting down, if you give something that's going to interfere with T-cell proliferation, at least during the early stages of therapy, that's not a good idea to give something like that. There is, however, we know that once the T cells finish the proliferative stage and go into the killing phase, you can block proliferation all you want, you know, so you just got to be conscious of, you know, what the drugs do and at what stage of a response. For small molecules, it's not a simple thing to do because it's hard to predict, since these drugs of that type, the entire clinical development until they start treating people has been done in mice without immune systems. And so, you know, with xenografts of human tissue into mice that don't have immune systems, and so the effects of those drugs on the immune system is completely unknown. They've never been in that situation, and you know, we just need to know that. On the other, and some of them might be good though. We did some studies with vimurafenib, which is a inhibitor of BRAF, and we found that it actually potentiates immune responses, paradoxically activates the MAP kinase pathway and activated T cells, and so that would be one that you could use together most times, but you wouldn't predict that from, you know, the fact that that particular drug is thought, you know, was thought to target the mutated molecule, because there's no mutated molecule in our setting when we did the experiments. But I think it's wide open. It just requires knowledge of the effects both ways.
Dr. Allison?
Sorry.
Dr. Allison, until recently, for prostate cancer, the accepted remedies were either excision or treatment with pellets. Until your research. Can your research be used on a prospective basis, preventative measure?
Yes, I've been asked that a lot. I don't think so, because you have to have sufficient antigen there to get a response to, and you know, you don't necessarily know what that is. Now, having said that, there are some situations where a big fraction of lung cancers are caused by mutations in a thing called epidermal growth factor receptor, and there's a stereotypical mutation there. And so, if you know somebody has they might already have to have tumor, but you could treat them very early. There may be some situations if susceptible targets could be found in breast cancer, you know, where there's a hereditary risk but I don't know what those targets would be. There are people now in Lynch syndrome, you know, which eventually can give rise to colon carcinoma. That there may be some promise there too, but you'd have to identify something that was shared by all the tumors, and you could vaccinate with people who had that trait, whose family had that trait. So there's hope, but I just can't tell you right now.
Hi, I'll have to preface my question by saying that I'm an economist. But I've just heard from a story from a colleague, and I'm wondering if you have any insights on it. Her 2-year-old was recently diagnosed with cancer, and she described a process where she actually had to have the 2-year-old injected with a lethal dose of the cancer and revived with stem cells. So I'm wondering, is there any theoretical overlap with a procedure like that with some of your research, and what are the underlying issues?
Yeah, there's a theoretical aspect. They're basically thought to be, for some cancers, particularly blood cell cancers. Well, there's stem cells, obviously, that can give rise, you know, to the whole population of cells, but they're also thought to be, in some cases, cancer stem cells, which already have the mutation that's going to, when the cells get to a certain stage, they'll start becoming cancer. So if you could identify, you know, that thing that makes the cancer stem cell, you know, carry the ability to, you know, cause the cancer, you could target that perhaps with a vaccine and checkpoint blockade. So people have talked about it a lot, but I haven't seen it reduced to success yet.
I read a fascinating paper recently where they found that cancer cells didn't have the circadian rhythmicity that normal cells do. Apparently normal cells take in nutrients on a certain pattern depending on the time of day. And the significance of this was if they challenged the mutations that stopped or allowed the cancer cells to feed continuously, it killed them. Have you heard of this approach, and has it been used in combination?
No, I haven't, but if there are specific molecules that are involved in that, certainly might be a possibility.
Do you want me to send you the paper?
Yes, please.
Will do.
Was it by Rick Scott, or was it from Rockefeller University? Oh, okay. Okay, good. Yeah, I'd like to see that. Looks like everybody got tired.
Thank you very much for a very interesting presentation. You've mentioned 2 or 3 times just in passing the possibility of treating breast cancer eventually. Could you elaborate on that?
Well, there have been some success with vaccines in breast cancer. Not great, but treating residual disease, and so there's a big effort going on in a number of places that are getting some responses. It's not quite there yet to the level of where we could say that approval by the FDA is imminent, but I guess I've just seen enough that it looks like we're starting to get there and get hints of responses that can be built on now. I can't get into the specifics, but it's promising.
Thank you so much, Dr. Allison. I think that was an amazing talk. I just wanted to thank you also. There's going to be a small break where we have lunch for everyone. It's going to be in the lobby as you walk through, and you can come back to the tables and have lunch. And then after lunch, we're going to continue our discussion with Dr. Allison. Thank you.